This video will cover the basics of protein structure, including the types of bonds that stabilize proteins, the levels of organization, denaturation, and an introduction to simple and conjugate proteins. Proteins are large biomolecules made of one or more long chains of amino acid residues. They perform many functions, such as catalyzing metabolic reactions, providing structure, and transporting molecules. A protein's specific 3D structure is determined by its amino acid sequence, which dictates its activity. Proteins are polymers of amino acids joined by a covalent peptide bond to form polypeptides. These polypeptides fold into a specific functional conformation. This folding is stabilized by various strong and weak bonds, including peptide, ionic, disulfide, hydrogen, and hydrophobic interactions. This section focuses on the bonds stabilizing protein structure. 5 main types of chemical bonds play a crucial role in determining and stabilizing the 3D protein structure. These are peptide bonds, ionic bonds, disulfide bonds, hydrogen bonds, and hydrophobic bonds. A peptide bond is a strong covalent bond formed between the carboxylic group of one amino acid and the amino group of another. This condensation reaction eliminates one water molecule and forms a dipeptide. A dipeptide can form additional peptide bonds with other amino acids, leading to a polypeptide chain. Peptide bond formation is facilitated by the enzyme peptidyltransferase during protein synthesis. The peptide bond is essential for stabilizing the protein's primary structure, which is the linear sequence of amino acids. Ionic bonds are chemical bonds formed between two ions of opposite charges. In proteins, they form between ionized acidic or basic groups in the amino acid side chains. Acidic R groups become negatively charged by releasing H+ ions. Basic R groups become positively charged by accepting H+ ions from the medium. The attraction of oppositely charged R groups creates ionic bonds, also known as electrostatic interactions. These are weak and easily broken by changes in pH, which can lead to protein denaturation. Ionic bonds stabilize the tertiary and quaternary structures of proteins. A disulfide bond is a strong covalent bond formed from the oxidation of 2 thiol groups from 2 cysteine residues. Cysteine contains a reactive sulfhydryl group in its side chain, which allows for this permanent connection, also called a disulfide bridge or SS bond. These are very strong bonds that can form within the same polypeptide chain or between different chains. Disulfide bonds are crucial for stabilizing the protein's tertiary structures. A hydrogen bond is an electrostatic attraction between a hydrogen atom covalently bound to a highly electronegative atom, like oxygen or nitrogen, and another neighboring electronegative atom. The difference in electronegativity creates partial positive and negative charges. The slightly positive hydrogen is attracted to neighboring electronegative atoms like the oxygen in the carbonyl group or the nitrogen in the amino group. This occurs regularly along the polypeptide chain, giving it regular shapes like alpha helices and beta sheets. Though weak, a high frequency of hydrogen bonds contributes significantly to molecular stability, stabilizing the secondary, tertiary, and quaternary structures. This image illustrates a hydrogen bond, an alpha helix, and a beta-plated sheet. Hydrophobic interactions involve non-polar R groups, such as those found in amino acids like alanine and valine. Since these groups are hydrophobic, they try to avoid water. In an aqueous environment, the polypeptide chain folds so that the hydrophobic amino acids cluster towards the interior, excluding water. This folding allows globular proteins to form a spherical shape. In a folded protein, hydrophobic groups are oriented inwards, while hydrophilic residues form a soluble shell over them. These interactions also influence the orientation of membrane proteins. Next, we will explore the different levels of protein structure or organization. Proteins have 4 levels of structure. Primary structure is the amino acid sequence. Secondary structure, such as an alpha helix, is formed from the polypeptide. This is part of the tertiary structure, which is the final folded polypeptide. Quaternary structure is the arrangement of multiple polypeptide subunits, like in hemoglobin. Most proteins fold into a unique 3-dimensional structure called their native conformation. While some fold unassisted, others require molecular chaperones. Biochemists define 4 distinct aspects of protein structure: primary, secondary, tertiary, and quaternary structure. We begin with the protein primary structure. Primary structure is the linear sequence of amino acids reported from the amino terminal N end to the carboxyl terminal C end. This structure can be directly sequenced or inferred from DNA sequences. In biological systems, proteins are produced by ribosomes during translation, where amino acids are polymerized via peptide bonds. Peptides can also be synthesized chemically in the lab, typically starting from the C-terminus, the opposite of biological synthesis. Protein sequences are typically notated as a string of letters, using either a 3-letter code or a single-letter code for the amino acids. Extensive sequence databases exist that compile known protein sequences. This image shows the notation for the 20 natural amino acids and ambiguous amino acids. Although polypeptides are generally unbranched, proteins can be cross-linked, most commonly by disulfide bonds. The primary structure must also specify these cross-linking atoms, such as the cysteines involved in disulfide bonds. The chiral centers in a polypeptide can undergo racemization, where the L-amino acids can spontaneously isomerize to D-amino acids, which are resistant to most proteases. Additionally, proline can form stable transisomers at the peptide bond. Proteins can undergo various post-translational modifications. For example, the N-terminal amino group can be covalently modified by acetylation, which eliminates the positive charge, or formylation, which blocks the N-terminus of methionine and is usually removed. Other N-terminal modifications include pyroglutamate formation from N-terminal glutamine and myristoylation, which adds a hydrophobic tail to anchor proteins to cellular membranes. The C-terminal carboxylate group can also be modified by amination, neutralizing its negative charge, or by glycosyl phosphatidylinositol attachment to anchor proteins to membranes. Phosphorylation is a crucial chemical modification. A phosphate group can attach to the hydroxyl group of serine, threonine, or tyrosine, adding a negative charge. This is catalyzed by kinases and reversed by phosphatases. Phosphorylation often induces conformational changes or allows proteins to bind to one another. Glycosylation is a common and heterogeneous set of modifications where sugar moieties attach to the side chain hydroxyl groups of serine/threonine or the amide groups of asparagine. These attachments can increase solubility or facilitate complex recognition. Deamidation involves an asparagine or aspartate side chain attacking the following peptide bond, forming a succinimide intermediate. Hydroxylation adds a hydroxyl group to proline or lysine residues, which is critical for components like collagen. Methylation occurs on positive groups of lysine and arginine. While methylation does not change the positive charge, acetylation of lysine regulates protein binding to nucleic acids by weakening electrostatic attraction. Sulfation of tyrosines adds a negative charge and is used for specific recognition. Prenylation and palmitoylation add hydrophobic groups to cysteine residues, anchoring proteins to cellular membranes, similar to GPI and myristoyl anchors, but not necessarily at the termini. Carboxylation adds an extra carboxylate group to glutamate, strengthening binding to metal ions like calcium. ADP ribosylation transfers a large ADP ribosyl group to various side chains with heterogeneous effects. This modification is a target for potent bacterial toxins. Ubiquitination and sumoylation attach folded proteins to the side chain ammonium groups of lysines on other proteins. Ubiquitin commonly signals for protein degradation. Most of these modifications occur post-translationally, typically in the endoplasmic reticulum. Peptide cleavage by hydrolysis or proteases is a very important modification. Proteins are often synthesized as inactive precursors, which are activated by cleaving off an inhibitory segment. Some proteins can even cleave themselves through a chemical reaction called an NO-acyl shift. The resulting ester or thioester bond can be resolved in several ways. Simple hydrolysis splits the chain, forming a new N-terminus. Beta elimination splits the chain, resulting in a pyruvoyl group. Intramolecular transesterification results in a branched polypeptide, while intermolecular transesterification transfers a segment from one polypeptide to another. This section covers protein secondary structure. Protein secondary structure describes the three-dimensional form of local segments of proteins. The most common elements are alpha helices and beta sheets, along with beta turns and omega loops. Secondary structure is defined by the pattern of hydrogen bonds between the amino hydrogen and carboxyl oxygen atoms in the peptide backbone. Alternatively, it can be defined by the regular pattern of backbone dihedral angles in the Ramachandran plot. Alpha helices and beta sheets are the most common secondary structures. Other helices, like the 310 helix and pi helix, are energetically favorable but rarely seen in natural proteins, except at the ends of alpha helices. Other extended structures, such as the polyproline helix and alpha sheet, are rare in native proteins. Tight turns and flexible loops link regular secondary structure elements. Random coil indicates an absence of regular secondary structure.